Hello friends, welcome to BMH Learning. In this video, we will talk about codominant and dominant markers. First of all, let's remind what is a DNA or genetic marker. A genetic marker is a gene or DNA sequence with a known location on a chromosome that can be used to identify individuals or species. It can be described as a variation that can be observed. Now, the variation may arise due to mutation or alteration in the genomic loci. The genetic markers are of two types, codominant and dominant markers. Or we can say the genetic markers can exhibit two modes of inheritance, that is, dominant recessive or codominant. Let's understand the difference between the two with the help of an example. Let's suppose you are working on a plant trait with two alleles. One is wild type, that is capital A, and another one is mutation form, that is small a. So there are three possible genotypes: capital A, small a, capital A, and small a. The first two are homozygous, and the third is heterozygous type of individual. Let's do PCR for the specific locus we are talking about using both codominant and dominant markers separately. So, when you do PCR in case of codominant, you can see bands for both capital A and small a. Therefore, for capital A and small a plants, you will see only one band. And for capital A and small a plant, you can see two bands. The advantage of this is that you can distinguish homozygous from heterozygous plants by genotyping. So, codominant markers can differentiate alleles of same gene. SSR is one of the best examples that determines allelic variation in population that can help differentiation of genotypes in population. Now, on the other hand, if we do PCR with dominant markers, we would observe totally different pattern. You would see bands of only one allele. Or in other words, this means one allele band is not shown in the gel. So wild form, that is capital A, is there, but mutant form, that is small a, is absent in the form of band. So this implies when you see the band of heterozygous having both alleles, we observe only one band specific for dominant allele, that is capital A. Now, the problem here is that the homozygous and the heterozygous can't be distinguished from each other, because in case of heterozygous, only dominant is seen. So this is why codominant markers are preferred for most applications. This was all. Thanks for watching.